to the second MMIV seminar of this year. So today we have two speakers. One is a long-time member of MMIV, and we have a junior member, so Hauke Bartsch and Tanja Eichner. And so we have two speakers, means we have ample time for questions and discussion. And these seminars are meant as an exchange as well. So you're invited also at home to ask questions and discuss. And so I will introduce the first speaker, Hauke. Let me tell a couple of words, especially for new members, and maybe hear his name a lot and wonder some more. So, is Tanya first? Is it?
No, no. I don't know.
We have exchanged the order with respect to the. So Hauke is one of the pillars of MMIV, or maybe more modern expression, one of the CPUs of MMIV, and many years of experience in the medical industry and academia, and is doing developing and coordinating a lot of very diverse projects here at MMIV, especially about the integration of machine learning methods in the clinical workflow. And indeed, today is going to present some work in progress about a very important problem in medicine, the fact that we often have to use data that are few and biased. And the title of the talk is Data is Code is Data. Can generative model replace data for image segmentation? Peace out.
So my name.
Yes, I know all of you, so but there is Zoom as well, and I just afraid to embarrass myself. So this is very unpolished. There's it's basically the things that I do for fun. So you have to bear with me. It's not as nice as the presentation you will hear later from Tanja, but at least it's with a German accent as well. So I try to, it's like to make things freshen them up a little bit. There is this you have to make them come to a point. You have to make it really, you have to stress the most extreme case that you can think of. And this is very much written like this. So it's not, really meant like it. But if you think some of the thoughts to the very end, then at least there is an argument you can do for half an hour just playing around with this thought that maybe that this is what we should do. And specifically, generative models just have a long history. So I've used them for many years, and I feel comfortable with them, and that's also part of why I'm falling back to those. So if you have if a different history, you might use other things. So it's totally biased. It's just my own opinion. There is nothing canon about any of this. But it has, I think, some beauty. So I just read the abstract that I wrote Luca when he asked, well, can you present something? So if we presume that medical data is sensitive and scarce and costly, imbalanced, the interesting cases, there are just few of them, and also biased, by the institution, by the specific data source that you have, then it is very dangerous to actually use them for training a model, because you have two unknowns. You have a model, you don't know what it really learns, and you also don't know what is really in the data. So these two unknowns, they can make it that you're lucky, everything works, you're fine, black box, forget it, done. Or you can be very unlucky. And who can be unlucky? You, if it doesn't work, because you can always blame it on the algorithm. But also the patients can be unlucky. There can be this one group of patients that just the model consistently does something wrong, somebody dies. This is medical imaging, we have to be careful. So I'm basically trying to argue to not use any data. So if we have to use data to train. we know that. We don't want to like redevelop how we learn, but maybe we can take data or develop an understanding, which is then the code part, and from that derive data that we use for training. So it's a little bit the devil you know. It's better than the devil you don't. And if you really understand how your data is generated, what it contains, maybe that's preferable in some sense over data that you don't know and where you just have to hope. Anyway, it's actually part of a couple of presentations. I'm just pulling a couple of slides together to get it into the context of the DIML project. So we do this project lifecycle management. It basically means that projects, they start and they end. There's lots of things that happen in the middle. And this type of image analysis is always done as part of a specific project. So you see here that there's lots of different steps that we all know and that are all part of the procedure. And basically, we're using a system that captures the whole workflow. I don't want to go into too much of a detail, but we have access to the structured data and to the image data for real problems. The data comes from like all of these different institutions. Scanners are directly connected, and they go through like a channel bottleneck that makes them all the same. So we can guarantee some properties on all of the image data, regardless if the data comes from prostate or from lung CT or from something else. So hopefully when we develop algorithms, we do this like AI, machine learning, deep learning, we do this on a data set that is so normalized that it's very easy to just take the same algorithm and apply it to a new context, to like a different project's data. It's like a drop. Usually that's what we want. The big divide on the right is all the data we have in the hospital in this nice system. On the left is all the open source software that we would like to apply, where we are setting up our own like laptops and computers to just do the analysis. Where really the data is, where the rich data is, where the data is that is actually relevant for practical purposes, is not on Kaggle, is not on hosted sites. It's actually inside the hospital. It's inside the research projects that we are supporting every single day. So the basic idea is very easy. We take algorithms, we put them into a container to make them movable, and then we move them into the hospital. And we have like a window into the hospital, so we can do this not just once at the end of the project, but as part of the development of the algorithms. So you start with a stupid prototype, you try it on data, you see the effects, you change it, you move it back in, you see what it does, etc. It's a little bit more complex than just have local copy of the data. And you can mix the two approaches, of course. But it will allow you to move it into the hospital, so have a radiologist view the results instead of you having to open a PowerPoint, pulling together some images. A couple of things that we do. The hospital data is in the clinical packs. It's moved into the research packs. We select specific image series based on some DICOM tags. We select the level of analysis. The analysis is either training or model prediction, longitudinal analysis, or just a cross-sectional analysis. And then we apply our algorithms that we have uploaded as containers to all of the data in the project. So very simple. Of course, the one unique thing that everybody knows how to use is web interfaces. So it's just a web interface that allows you to implement the workflow. We started with uploading the container. There is a setup where it helps you to structure the container. We have a couple of people who are going through this right now. We're trying to make it easier. You get the permission to access a specific project. And that means that your algorithm is allowed to see data from that project. And then you can just run it. So it's, it's inside the study. You have a way to structure what data is visible to the system. This is what it looks like. Just a reminder, it's the selection of image series and the selection of the level of analyses. Those two come in two steps. It's very simple. It's just 3 lines of code here. It's like repeated 4 times. This is how you do prediction on a single image series, and this is how you do model training on a whole project's data set. So it's like really easy, and it just appears on the user interface as part of the model setup because it's specific to the project. What is a T1 in one project is different from a T1 in another project. That's why it's belonging to this one. And then you press the magic button, runs everything, and it will have like an output by job. What it's doing here is adding a little bit of visualization to the produced outputs. This particular run is using MRIQC. And MRIQC is a container that generates maybe like, I don't know, 50 to 100 measures for signal to noise, all kinds of segmentation results for a particular type of MRI image data. And they all come back. So each of the jobs that produce then lots of structured data and tables and new images, and all of them are sorted according to how uniform are they in that data set. You can see here like a random score that is assigned, and if the score is like the same, then you can kind of assume that it's similar data in terms of MRIQC. And if the score is very different, I think it's like further down, there's like 190% score, then you should look at the data. That is a functionality that is independent of the actual measures that you have. That's why we like to have the web page. So regardless of what Docker container you move in and analysis you do, you're always producing some structured data, and therefore you can do something like this. It's an algorithm that stretches across all the different projects. Okay. How to learn now without learning from actual data. So there's a couple of things that I mentioned, like the pseudonymization, making the data all look the same so that your algorithms apply across projects. That has some level of error. If you're anonymize something or pseudonymize something and have the data useful. Image data, I think you can ask a radiologist and they tell you, oh, somebody could recognize the face, so you do face stripping. That removes large parts of useful information. Some other people will tell you, yeah, the ears are actually identifying, so you should reduce the ears as well. And you can easily show that, in Alexander's data, for example, that the folding patterns of the brain, they're also identified. They're also a fingerprint because you can see which brain belongs to a person just by looking at the folding patterns. So if you go to the very end, you basically have a trade-off. Either you remove all the information in the data, and then it's really secure and you can't do anything, or you go kind of in the middle and you have to know, what is the current state of art. So how much of the data should we remove and how much of the data we require for further processing. But that means that there is some percentage of error that is left. The biggest projects that we will be doing in the near future is like a mammography project. It's 150,000 participants. So we will have data from 150,000 participants. So even if this is like a factor of like 100 smaller, the chance of having an error is still not zero. And just with scaling to larger projects, you will end up with problems. Yeah, human subject data. I was basically just trying to make some points to be able to write the sentence down here. So it's like, so we need, of course we don't because we can still do work, but we need 100% secure and balanced way of model training without image data. That's the dream, isn't it? If we just can write to the ethics board, this algorithm has actually not been, there is no patient data in danger because there is no patient data in it. And it is not a black box. what it learns, because we know exactly what data goes into the model, we would just we don't have to do a rec approval because it's basically the sensitivity that is in the patient data that we take out of the equation. The question is, can we do anything useful if we really do this? That we have to show. I can't really show that we do this in this talk, but I'm kind of working on it. So 100% secure and balanced way of model training without image data. It's pretty easy. People have tried to limit the dangers. So for example, they use publicly available data, and I think that's just garbage. It's useful, but it doesn't solve any problem. So one of the issues is that in the public, people are very much aware of AI techniques. So if somebody comes into the hospital and we ask them, would you be willing to participate in a research project? They will just look at the latest news and say it's like, oh, there's all these data breaches. No. We don't have any image data anymore in the future. So we have to maintain an image towards the public where we say it's like, it's not really dangerous. We take care of it to the best of our knowledge, and we implement all the latest things in it. So using publicly available data doesn't mean that you're off the hook. It just means it's other people's private data. That means there's real patients there, and there is a backlash that will reach you if you're using it wrong. So let's say you go to a publicly available data set, download, and then detect the ears and go to Facebook and then try to re-identify the person. This is like the worst thing you can do ethically. Even showing that that is possible is not useful, because it would basically, it's like spend your time to cure cancer instead of just breaking into systems or show to break into systems. What is happening right now with respect to data access for these publicly available data sets is that data access is successively removed. So these publicly available data sets, there is put a barrier in front of them. And the barrier is using institutional membership. So as a private person, you will not have access to these data sets anymore, but you will have only access if you're part of a big institution. The institution has to sign for you that you will not reuse the data for nefarious purposes. So you will basically lose your job if you re-identify data, try to re-identify data. It will also happen that the structure is kind of, instead of getting the data and then it's yours, you will get only a loan of the data. And I think that's very appropriate. It is something, every year you should reapply and say, it's like, I would like to get continued access to the data. And that's because we want to be able, if some of the participants come and say, it's like, I don't want to have my data used, then we should be able to implement that. And that can only be implemented if the data is centralized, if you get access to it for a given period of time, and then data can change. You can remove stuff. So that will generate, basically, that's kind of an outlook into the future. Generative adversarial networks, difficult to train, really interesting, good for style transfers, but it's this Münchhausen kind of, I didn't find a picture. It's like an old German story of this guy who is in the 100-year, 30-year war. I don't know, long ago. So he is just riding on his horse, and there's a swamp, and the horse is sinking. So he is getting himself out of the swamp by pulling on his ponytail, and then he is like getting on land again. So that's kind of the story, and replacing data with data that comes from in these adversarial networks from patients, basically it's just like a rewording of the data. You will get pure less quality data in the generative adversarial networks, and there is no additional information that the network can actually put in there in theory. That means you will only see the data that has been used to train these adversarial networks, and they will leave an imprint. So if there is an imbalance in the training data used for a generative adversarial network, that imprint will also be in the generated data that comes out. I like this one really. Do you know the Dolly, this project? You probably have seen the picture. There's this avocado armchair. So it's basically a network. Everybody is working on this right now, combining text and images. So you can give it an image and it will give you a description, and you can give it a description and it will give you an image. So the description, the famous one, is an armchair in the shape of an avocado. That's the text. And it produces really nice images of avocado armchairs that look like 3D and really relevant. And so it's basically generating an image from text. So that's the dream, isn't it? We type in cervical cancer and we get an image of cervical cancer. Then we know it's like that's the image that we can use for training, and the only thing that went in there is the text, if that's really true. Okay, so I will go with this last one. Of course, this is a talk about a particular project, so we are using particular image data. This is malignant. There is lung segmentations, lesions that are from cancer cases. You see an example image here. It's CT, pretty high resolution, 3 mm slice distance, like 256 by 256. It's a publicly available dataset, LIDC IDRI. And there have been different raters who just went in the data and annotated the big lesions by drawing polygonal shapes. You can see two of them sitting on top of each other. So the data is available as a dataset in the research packs. So everybody can just, because it's publicly available data, just go to the website, fill out the form, send us the form, and then you get access to this data on the research packs if you want to use it. So you see the workflow here. It's basically pull the largest index lesion and then measure the volume. So this is what it looks like then on the algorithmic end. We start here on A, on the very left. So this is one of the images. It's 3D data, but it's a 2D slice. And the algorithm implemented is just like getting from that to like 3D stack of images to a 3D model, like a surface representation of left and right and the airways. And the way this is done is very easy. You basically just, because it's CT, you know the intensity range, so you can like label everything above air as body. And then you fill in everything. And then you have like a solid body shape. And inside that mask, you look for everything that is air. And then you get only the lungs and the airways. And then you go to the very top slice. And in that top slice, there is no lung. The data is acquired like this, but there is like the airway sticking out. So you can see inside the body an airway that is a single object. And that's then, by definition, the airways. And from that segmentation in 2D, you go to the next one. Connected components, next one, connected components, next one. Until you're in the airways, and there it divides to the left and right lung. So you know that the surface area of the airways gets a little bit bigger and different objects. So you know then to stop. And then everything else is lung. And then if you're on the top of the lung lobes are very well separated. So you can basically see there is one object, and it's because it's left and right. You know, this is the left lung, this is the right lung. And only in the lining here at this place, and sometimes down here, these two lungs will touch. So you can use morphological erosion to make sure that they're separated until they're two objects, and then dilate again to fix them back together. And then you will have 3 labels. You can see an example here what the module produces. There is the data set like this, which is just a lung tissue. there is a data set like this, which is a fused image of the 3 different labels. And then, of course, what is not displayed is just the labels on its own as like a lifty or a stack of DICOM images. With this information, you can go back to the original data, extract all the lung tissue, do volume renderings, anything you like. The picture up here is actually from a textbook. What they did is take a lung out and fill all the vessels with raisin, and then it's removed all the soft tissue, and then you get these trees, and you see the branch points, the blood vessels. They separate the lung into the different lobes. That's basically, this is trying to do the same. Okay, so step 1, we have the lungs. Step 2 is now, so the lung tissue we can get out from real data set, but we want to train now a classifier which will just take tissue and say that lungs, their blood vessels in there in different directions. But sometimes there is this object in it, which is the cancer we are interested in. And in this case, we are not using the data that we have been provided, but we are replacing it with the idea of the data. And our idea is there's like vessels, and sometimes there is something in there. And we generate lots of vessels with objects in it. And we generate them, and that helps us to actually train a model that on this lung tissue will be able to recognize elongated vessel-like structures and distinguish them from floating objects that are in the middle. You can see in an example over here. Oh, there's even a movie. Okay. We'll try to switch to okay. So I hope you can see this. Just a couple of steps to get from noise to vessels with lesions. And I've showed this before. We'll just go to some steps like 1D, and then you can extrapolate to 2D. So here's an example, random values around 0. 0 is the horizontal line, and there is just independently sampled random values along X. And if I low pass filter this, then I basically, like the Gaussian moves through, and the data becomes more similar. So if there's a high peak and the next one is low, then afterwards it's like kind of both are in the middle. And that means that what was like independent samples before now become correlated with each other. And the more often I press this low pass filter button, the more smooth the whole thing becomes. I'm showing them as bars, the numbers, and I'm showing a switch from a negative value to a positive value with a dot. And you can see here 1D data, the crossing from positive to negative, the sign changes are dots. It's just dots in space. You can do this once again. You can see it becomes more and more smooth if I do this, which means the points move further away from each other. And I can do this twice. So it's like the first one are the blue ones, the second one are now the yellow ones. They're done independently. So I smooth the blue ones, I get red dots, and I smooth the yellow ones, I become other colored dots. And in 1D, these dots, there will be like random positions, so they are not overlapping. But what I can do, just like, what is happening? Yeah, here you see the crossings of the blue one. Now I just do the same in 2D. So instead of a 1D array of dots, I have a 2D array, and I have positive, negative values randomly. And now I fold it with a Gaussian. So it's random. After one iteration, there's smoother structures, 2 iterations, 3, 4, 5, 6. You see what happens. The number of iterations is basically related to how big the structures are. So I can have like very small structures if I don't iterate a lot. But if I'm iterating like 20 times, only like very big structures will survive. And this is just a reflection of the fact that if you do independent random, you will get all frequencies in the data. There's like small frequencies and big frequencies. Folding with the Gaussian is like a low pass filter, a low pass filter that will pick out the larger proportions of the data. So this is nice because now we can do our zero crossings again. So it's like 1D zero crossings are dots, 2D zero crossings are lines. It's basically just one dimension less. 2D data, this is now like 1D lines, and in 1D it's zero dimensional dots. And you can scale this to 3D. So if we do this now in 3D, I have random. I'm not showing the values anymore. In 3D, it's difficult. I'm only showing the zero crossings. So zero crossings in 2D are lines. Zero crossings in 3D are surfaces. So you have one surface. On the outside of the surface is the high value. On the inside of the surface is the low value. And if I smooth those, you can see there's two different colors, yellow and blue. So the blue one is the one field, the yellow one is the second field. 5, 6. Okay, maybe that's good enough. Really not nice looking, but I have some boxes floating around, like these red boxes. They're over here as well. These boxes are basically where the two zero crossings intersect. And the reasoning for this is I have two independent fields. There are surfaces, 2D manifolds in space. Intersection of 2D manifolds is one dimension less. So instead of getting surfaces, I'm getting line segments. And that's exactly what I want. I have a cube of data, and I have line segments in the cube that are purely generated from random noise, from white noise structures. I can control it with a given resolution by the number of iterations. And that means I have now, these boxes that connect in space along lines that are formed by the zero crossings of two independently, etc., etc. You get the gist. Okay, and that's the whole algorithm. What can you do with it? Here's an example. So if you do this then in 3D, you can shift the zero crossings. So there's like two parameters because you have two surfaces, you have two offset vectors. So you can generate not just one set of lines in 3D, but you can generate connected sets of lines in 3D that will be guaranteed not to intersect. And they're just like a little bit offset from each other. So here it's just 3 of those generated. You can see a 2D version of this. So of course, the whole thing is done discretized. So I'm starting with a volume of a given dimension. You can make that arbitrarily big or small. And you can make that a real cube that is then matching to the resolution of the data sets that we get from the lungs. So you can basically match the spacing, the thickness of the lines, and the lung tissue using the arbitrary scaled version of this. It's just the same, surface rendered. There's even more information in there. This is an example where, because two independent random fields, they have the sign, so they have regions of positive and negative sign. And because you have two independent fields for each voxel, you end up with 2 sign values. So you have actually 4 different combinations of sign values that are guaranteed to be a specific distance away from any of the zero crossings. And that means you can color them, and you can say it's like I have a green region, and then whatever the color names are. I'm bad with naming colors, but you have spaces intermittent with the lines that you can fill with cells or fill with something else. It's basically just a logical subdivision. We are only interested in the lines, and we will remove everything else. Okay, going back to the presentation. That process that is here on the web page is basically just the ITK program. With ITK, you can do all the foldings and specifications, etc. And then it's just a loop. So I have a for loop and generating a sequence, 25 million images, volumes in this case, that are all guaranteed to be different because there's different random seeds. I can generate as many of those as I want. And I can randomize some of the parameters. I could say it's like, the sizes of my lesions are small or big. I know where to put them because they go into these green large object regions. So I can control where my lesions will be in the data. Are they free flowing? Are they attached to one of the vessels? I have a choice because I know what is close to a vessel. I know what is far away from a vessel. So I can basically place them, just doing some random sizing of lesions, ellipses. Okay, so you end up with folders and you just count them up, use them as training data. Training is really easy. You just use MMIV because it's 3D and the whole code is this long. So you saw data, this is like 90% code for training 3D is really easy. And it works fairly well. The model does something after a couple of iterations and it's predicting. I can do the same with cells. It's like 3D stacks of cells, very easy. So usually this is what the data looks like. After you get it like from a confocal, you do a deconvolution, and then you do a segmentation, and then you have like the outlines and the dark dots and the cell walls, and that's basically very, very time consuming because you have to do it in 3D, but we can just turn it around. We start generating cells, and then we're adding just a convolution with the Gaussian that has like X, Y, Z components. And then we can add some noise, and then we have image data that looks like cells. Here's how this works. Start with an image volume, put some points in, use a Voronoi tessellation, and then mark the different objects as independent cells in this case. And you can see that where they intersect because of the Voronoi tessellation, there's a nice border between them. And then you do edge detection, and you just get the outlines. Then you know exactly which voxels are the cell walls. And then you add inside the labels, you know what the inside label is. You add just at random positions objects that could be the cell cores. Very straightforward. Again, because you're generating this from the label field, that's where you start, and then you make it an image, you can guarantee that there is perfect separation of cells. There is zero overlap, there is zero touching. Here's an example of like one of these cell configurations. If you do a surface reconstruction, it's exactly the data as you would like to see it. You can then export this as individual images, and that's like 1,000 cells cores for like 100 slices each. It's millions and millions of data you can generate with this. It's an example generator. This is what it then looks like in MMIV. So just as an outlook, what is coming, the generation of lots of these measures, we all put them back into RedCap. There's actually a data exploration portal that will organize the data, all the different measures that you see. And it's directly connected to our RedCap structure, allows you to search the definitions of the algorithms that are used to generate the data. You can run statistical analyses inside the portal as well. So you can then immediately say, it's like one of the columns is the diagnosis. And then you have a lot of choice about covariates of no interest and covariates of interest. And you can test this directly on the model on the web page.
So I think
how many minutes was I over?
Okay, I think we'll save his questions till afterwards because we need to move on to the next speaker. Next speaker is Tanja Eichner. She is studying medical informatics in Vienna, but she's currently taking her master thesis with Nusra and Eric here at MMIV. And today she will present the current state of her thesis, which is about an interactive co-registration application for MRI data based on segmented tumor masks. And this kind of application would be super helpful in my work. So I'm very excited about Tanja's research.
Yeah, thank you. Thanks very much for the introduction. So Tanja already introduced me, so I can go straight to the content if I can click here. No, yeah, okay. So I'm working with endometrial cancer, and as in many other types of cancer, you acquire multiple sequences per patient. Why do we do this? We see different features of the tumor in each of the sequences. And one current hot topic in research is radiomic tumor profiling. So from all of these images from one patient, we try to extract different meaningful features in order to potentially define new biomarkers for tumors and characterize one specific kind of tumors. So this would be the goal, but what do we have to do to go from the acquired images to perform radiomic tumor profiling? We basically need two major steps, let's say. One is we need a segmentation, which is that we need to see, okay, where is actually the tumor in our image, and have an outline of it, like this red mask here, for example. And the second thing is registration. Why? Let's say if we get the images, we can't guarantee that if we overlay them, that the tumor is always exactly at the same place. Even though in theory, like the patient is lying in the scanner and does not like move a lot, but still like we have internal organs which we can't control. So in the area of the pelvis, where also the data is acquired from, there's, for example, the bladder, which changes the filling. In the back are the intestines that are doing their work. So we can't like guarantee that the position is exactly the same for the tumor. In an ideal scenario for radiomic tumor profiling, it would go away like this. We acquire the images, we perform an automatic registration, like an alignment of the images. We segment this, for example, with like Erlen's and Satie's algorithms, and we can perform radiomic tumor profiling based on those masks. The reality looks a bit different because often the co-registration is hard. I will show you also why, but it's difficult. So what is actually happening is that still often, for example, Kari and Njol are sitting there and they're like outlining the tumor for at least one of the modalities. So we can work with that. And that co-registration is actually a problem. I could show that also when I was working on a project here in 2020. We tried to subdivide the tumors in different regions. And there we first saw that actually, yeah, the co-registration could be a problem. Because if we try here, for example, like, don't worry about these things, but if we try to divide the tumor, which is this bright structure here, below the mask, into two regions, for example, then instead of getting actual two tumor regions, we get a division like from the background and inside the tumor. So this is an example where the mask doesn't fit, and then this could produce also wrong research results or not accurate ones. What can we do to improve the registration, or what is actually currently available? There are a lot of sophisticated command line or automatic tools that can work well in some cases, especially CT is great. I think you don't have to be a radiologist to see that this is maybe not correct. I mean, I tried different tools with like default settings. They produce like completely different results. You can, of course, like adapt the settings and make it as good as possible, but still, in many cases, it's like the quality is not sufficient and we don't have the tumor at the same place. If automatic doesn't work, you usually go for user interaction. You can, of course, kind of try to manually align the images. For example, ITK Snap has a functionality for that. I had to use it when I was working in Vienna. It was hard. Like usually MRI data is often depending on the sequence, it's noisy. The contrast between tumor and the endometrium or the uterus is often not high. So it's difficult to align them by the volume. And then you focus on bigger structures like here the bladder. But as we said before, the bladder alignment is not correct because it can change the filling. So then you again have an incorrect alignment. What is often done in tools is that they use landmark approaches. So radiologists sit there and put, for example, too many corresponding landmarks into different images. This works also great for anatomical structures like bones, where you have predefined landmarks. You know, you set this point there and it's there in the other image. But for pathological data, you don't have that. And it's often difficult to see the outline. The tumor can change also a little bit, like the boundaries, for example, depending on the sequence. So we often don't have those clear anatomical points. And another disadvantage of all of these tools is that you process always just one modality at a time. So you have one reference modality, you load the other, you align it to them, and then you have to do it for all of them in a row. So that's where our tool comes in. So it's called MUSIC, which stands for multi-sequence interactive co-registration. And we want to make use of what we have. We have one segmentation mask that fits always one of the sequences, which in our case is the VIBE sequence. And we assume that the shape of the tumor between our scans changes only in rotation and position. Why do we assume that? Because the tumor in this area is the hardest structure. Like it has a lot of condensed cells. So before the tumor actually changes really the shape and not only like a bit the location, everything around it would change the shape. So, and we want to achieve that by letting the user, instead of the whole volume, just control the segmentation mask in a web page. And yeah, as an advantage compared to the other tools, we do it for all the sequences at a time. So we basically have a workflow of 3, let's say, major steps. So since we want to find like a tumor structure in the different sequences, what we first do is try to find it automatically. If that doesn't work for some of the sequences, or if the user is not completely happy, they can make, there's a user interaction to make adaptations to the position. And if the user is happy with the location in all of the sequences, we can align them based on the new position of the segmentation mask. I will not go too much into detail in the different steps, but try to explain the concept, approach we're using behind it. For the automatic matching, basically, first we have to do some pre-processing because often we don't have a good contrast like in the around the tumor. So we want to make the tumor border stick out and have a higher contrast. So here, for example, the bright lines, they just indicate that we have a high contrast. With that, we know the shape of the tumor because we have one mask, and we kind of imitate what the user would do. So we rotate it and translate it and see where at the border do we get high and similar contrast values. That's a theory. Trying that in every combination would take years. So we use an algorithm that which is called simulated annealing in very, very short. It applies some random rotation and translation. So if the tumor mask starts here, but then it jumps maybe there and it rotates a bit and sees could it fit there. If it's better, it stays there, tries to optimize it, and otherwise it goes to another location, roughly. And with that, we try to find the real position of the tumor. As I said, because the contrast is not so good, it's not always easy, so it might fail a bit. And to account for the user interaction, we need to think about, okay, we have basically a 3-dimensional data because we have like the slices in a volume. But our screen is 2-dimensional, so we need to think about, okay, what do we want to display? Because even if you see like a cube on a screen, it's only virtually 3D, but you see a 2D projection of it. So what do we see on the screen? In the web, because we do a web page, there's a WebGL that has shaders that are programs in which you can calculate basically what color you render for each pixel on the screen. To calculate that, so we have like this cube, which represents our data. And we basically, what we want to project is always an intersection of a plane with the cube at a certain position. So we can, this is like the typical view you see when you slice, for example, through a volume, you always have one slide. So this is now, let's say, a view from the oblique actual view. We usually acquire the data, and in this case, but we also, to make it a bit easier to rotate something in a 3D space, actually, we of course want to show it from the other side, like basically from top and the side as well. So we have like a 3-plane setup, and we project the intersections with the volume on the screen. For the interaction itself, they of course depend on each other. So if you have like 2 planes and you rotate one, then of course the other has to rotate as well and reflect the changes. And that's also very roughly, if you're interested, it's very interesting. I didn't know so much of that before either. And I don't know, Jitka's not here today, but he helped me a lot. So it was super cool. Yeah, and I will show you the interaction later in the video as well. So for the actual registration, if the user transforms the segmentation mask, we get a transformation out of it. We also have, of course, the coordinates in the volume, like X, Y, Z positions of the mask. And we use that transformation to also transform the coordinates. And this information is then used by a tool called Elastics that takes like one sequence which should stay fixed, like this is our correct one, the VB one, the other one that should be aligned, and the information about the different coordinates of the segmentation. And it uses that information to then align the volume based on the new position of the segmentation mask. And yeah, so this is how it currently looks like. Did it start? No. It start now? Yes, okay. So basically there, if you open the tool and the data, you have like a toolbar, you have the main view, which shows like the data from the 3 different angles, and the other modality views. You can interact with the slices, like scrolling, you can adapt the opacity of the segmentation, you can zoom in and out, like so you have a better view on the tumor. And so in theory, in the beginning, when you open the data, you would call like the automatic shape matching and see a proposition of the segmentation mask. We don't do that. We want to process it on our own now to see it. And the user can basically move around the segmentation like with the keyboard, rotate it a bit, check like it works depending on where your mouse position is in the different views. If you're not happy with it, we can go back because sometimes you get a bit lost in a 3D rotation. Yeah, and then we can do this for different sequences. Some of the sequences have the same biological basis because they're derived from one modality. So in order to make it easier to process them and put them maybe next to each other, we have like a customizable layout, which can help like to have bigger view or not, and also to group those sequences. So if you make adaptations in one, it gets adapted in the others the same way as well. Yeah, so if you're, let's pretend we're now done with the adaptations. You have like, this is the Viva modality, the reference one that will stay fixed where the segmentation mask fits. This will stay in the main view, and then you can call the registration, and all these sequences here will get transformed to this sequence, and then it will update on the screen, and you can directly check if it was successful or not. And in the worst case, you can, it's usually not necessary, but in the worst case, you can still modify it and run it only for one of them again, because the ones that are not transformed, they will just stay the same. So yeah, that's the current state, but we have, of course, further steps, like there's always a lot to do. Next week, I want to do a user evaluation. So whoever is like interested to try it, it would be very, very great and helpful. It's for now, like I mainly developed it for use for radiologists, because I think it can be kind of tricky to imagine like the 3D segmentation moving in a volume, which is like a bit easier if you have the background. But if you want to try it, it's still very useful information for me also, like regarding the interaction and everything. Apart from that, the automatic shape matching depends a lot like on the case. Like it's not very generalizable currently, and it sometimes works well, but since we have random components in it as well, it can be that it's off, and it's not a big problem. Like it doesn't imply the program to work, because the user can adapt everything, but it would be nice to improve it a little bit to have a bit of more stable approach for that. Yeah, we would like to work on a publication for that, and then hopefully make it also usable for CARI especially, and in general at MMIV. So yeah, that's basically of it from my side. I have some discussion points that came up in the last days. For example, as I said before, we can move around the segmentation, and biologically, it is only like small rotation and shifting, but because of imaging physics, it can still be that the segmentation mask doesn't fit perfectly, or it seems like it doesn't. So we don't know the actual borders of the tumor. We just define it based on one sequence. So we don't know what is actually like the truth behind it, and how should we behave if in one of the sequences it does not fit perfectly. So that's hard to tell. And the other thing is that, of course, like we can perform rigid registration. Rigid is only rotation and translation. But maybe for the future in radiomic tumor profiling, it might be helpful if also some of the surroundings, for example, the uterus, is aligned, because then you can extract also like surrounding information and see if that's helpful to characterize the tumor. But then, of course, we introduce some uncertainty as well if we make it deformable, because you make up information that is maybe not there. So you don't know if it's 100% correct what you do, because you interpolate between, like you have two voxels, but if you rotate them and it has not the same distance anymore, then you make up information that is between. And that happens, of course, also if you have different slice thickness and stuff like that. But that's something that we should keep in mind and think about, if it's better to only have rotation or translation, depending on the application, or also deformable. Like both works, but it's just a question of what is more suitable. So yeah, then thanks for listening. Thank you very much for the attention. And yeah, if you have questions, discussion points, feel free to.
Due to time issues, I think we need to wrap it up. But please just sit, and we will continue the discussion and questions over waffles, because Ellen has made tons of waffles. So the next seminar will be held 6th of May. That will be a personal seminar. Thank you to Tanja, thank you to Hauke for excellent talks, and See you all next time.
Thank you.